Hello students, today we are going to see in Biobotany third lesson chromosomal basis of inheritance. In this, sex determination in plants and mutation in mutation types, mutagenic agents and their significance. Sex determination in plants. Sex determination in plants, about 94% of all flowering plants are monomorphic. That means that male and female plants are separate, and 6% of same. Monomorphic means same. 94% of flowering plant, the monomorphic means that male and female stamen and carpels are present in the same plant. Some of the flowering plant which have that 6% of flowering plant only dimorphic, that means the sexes are separate. C. Allen discovered sex determination in plants. Sex determination is a complex process determined by genes, the environment and hormones. Sex determination in Saline latifolia is controlled by three distinct regions in a sex chromosome. Y chromosome determines maleness, X specifies femaleness, and X and Y show different segments. Sex determination in papaya. Sex chromosomes in papaya, Carica papaya, 2n equal to 36, that means chromosome. Papaya has 17 pairs of autosomes and 1 pair of sex chromosomes. Male papaya plant have X and Y, and female plant have XX chromosome. In papaya, sex determination is controlled by three alleles. They are small m, capital M1 and M2 of the single gene. Genotype in tabulation, that genotype for the papaya, the in tabulation genotype dominant or recessive, modification sex. Small m to small m if it is a homozygous or recessive, the modification is restrict maleness and it gives a female plant. If the genotype is capital 1 and small m, it gives the male sex, because the dominant is a heterozygous and the modification induces maleness, so it gives the male. And capital M2 and small m gives the bisexual, but it is a rare type. And capital M1 or M1 or M2 or M1 M2, which are the sterile type. Sex determination in Sphaerocarpus. It has an 8 chromosome. The diploid sporophytic is always a heterogamete. 7 autosomes are similar and in both male and female gametophyte, but the 8th chromosome of a female is X, which is larger than the 7th autosome, and the 8th chromosome of male Y is, which is comparatively smaller than autosomes. The mesospores with the X chromosome produces female gametophyte, and those with the Y chromosome produces male gametophyte. Sex determination in maize. Zea maize is an example for monoecious. The terminal inflorescence, which bears a terminate floret, develops from shoot apical meristem called tassel. The lateral inflorescence, which develop pistillate floret from axillary bud, is called a ear or cob. A substitution of two single gene pair, BA for barren plant and TS for tassel seeds. Gibberellin play an important role in the suppression of stamens in floweret on their ears. In this tabulation genotype, whether it is a dominant or recessive, modification, sex. BA, that is a barren plant. TS means tassel seeds. That barren plant BA or TS gives the double recessive, so it gives the rudimentary female. And in the genotype BA or TS plus, which gives the male sex. And BA plus or TS plus, that male and female, they are double dominant. They give the monoecious plant. And barren BA plus or BA plus and TS which give the normal female. Now mutation. Mutation means a sudden change in the genetic material of an organism is called mutation. Mutations are two types. One is a point mutation and another one is a chromosomal mutation. An agent which are responsible for mutation are called mutagens. The production of mutant through exposure of mutagen is called mutagenesis, and the organism is said to be mutagenized. In this tabulation, basis of classification, major types of mutation, major features, origin, whether it may be an spontaneous or induced type. Spontaneous occur in the absence of known mutagen. Induced occur in the presence of known mutagen. Cell type, if it is the mutation takes place in somatic, it is occur in somatic means occur in non-reproductive cell. Germline occur in reproductive cells. Effect on function, loss of function, if it is lost, eliminate the normal function. Hypomorphic means reduce normal function. Hypermorphic means increase normal function. Gain of function expressed at incorrect time or inappropriate cell. Molecular change, nucleotide substitution. Transition means a base pair in DNA duplex is replaced with a different base pair. Base pair means purine, pyrimidine. Purine is replaced to purine, that is adenine, guanine. Adenine changed to guanine, or pyrimidine to pyrimidine. Thiamine changed to cytosine. Transversion means in base pair purine changed into pyrimidine or pyrimidine changed to purine. Insertion, one or more extra nucleotide are present. Deletion, one or more nucleotides are missing. Effect on translation, silent, synonymous, no change in amino acid encoded. Missenses, non-synonyms, change in amino acid encoded. Nonsense, termination, create translation termination codon. And frame shift, shifts triplet reading of codon out of correct phase. Types of mutation. It's a point mutation and another one is a chromosomal mutation. In point mutation, it refers, point mutation means alteration of single base pair of DNA or a small number of adjacent base pair. Types of point mutations. In types of point mutation, Addition or deletion mutation are actually addition or deletion of nucleotide pairs. Nucleotide means purine or pyrimidine, are also called as base pair addition or deletion and collectively called indel mutation. Substitution mutation or indel mutation affects translation. Synonyms, we already saw the synonyms. Synonyms are silent mutation, one type, and missense or non-synonyms mutation or nonsense mutation, and frame shift mutation. Frame shift means a complete loss of normal protein structure and function are called frame shift mutation. Mutagenic agents. The factor which cause a genetic mutation are called mutagenic agent or mutagen. Mutagens are of two types. One is physical mutagen and chemical mutagen. Muller was the first to find out physical mutagen in Drosophila. Physical mutagen. Using temperature, radiation such as X-ray, gamma ray, alpha rays, beta rays, neutron, cosmic rays, radioactive isotopes, ultraviolet rays as physical mutagen to produce mutation in various plants and animals. Temperature, rise in temperature break the hydrogen bond between two DNA nucleotides, which affect the process of replication and transcription. Radiation, electromagnetic spectrum contains shorter and longer wavelength rays. Ionizing, they are classified into ionizing and non-ionizing radiation. Ionizing radiation are short wavelength and carry enough higher energy to ionize electron from atom. X-ray, gamma rays, alpha rays, beta rays and cosmic rays which break the chromosomes. And non-ionizing radiation, UV rays, have longer wavelength and carry lower energy. They are used to treat unicellular microorganisms, spores, pollen grains, which are possess nucleic located near surface membrane. And Sabarmati sonora, castor arona are some of the plants which is takes place. It's a mutant variety. Sabarmati sonora is a mutant variety of wheat and castor arona is a mutant variety of castor. Chemical mutagen. Chemical which induces mutation are called chemical mutagens. Some chemical mutagens are mustard gas, nitrous acid, ethyl and methyl methane sulfonate and ethyl urethane, manganese salt, formaldehyde, eosin, etherocin, and nitrous oxide alters the nitrogen bases of DNA. Co-mutagens, the compounds which are not having own mutagenic property but can enhance the effect of the known mutagen are called co-mutagens. Now chromosomal aberration. Mutation are two types. One is point mutation, another one is an chromosomal mutation. Chromosomal mutation means the genome can also modified on a larger scale by altering the chromosome structure or by changing the number of chromosomes in a cell. They are termed as chromosomal mutation or chromosomal aberration. Change in chromosome number. The chromosome number of somatic cells are changed due to addition or deletion, elimination of individual chromosome or basic set of chromosome. This condition is known as numerical chromosomal aberration or ploidy. Ploidy involving individual chromosome within a diploid set, aneuploidy. Ploidy involving entire set of chromosome, euploidy. Aneuploidy, it's a condition in which diploid number is altered either by addition or deletion of one or more chromosomes. Organisms showing the aneuploidy are known as aneuploids or heteroploids. They are of two types, hyperploidy and hypoploidy. Hyperploidy, addition of one or more chromosome into diploid set of diploid set of chromosome are called hyperploids. Trisomy, it is divided into three types: trisomy, tetrasomy, pentasomy. Trisomy, addition of single chromosome to diploid set, is called simple trisomy, that is 2n plus 1. And in Datura stramonium, it is an first reported by Blacksley, and but in later it was reported in Nicotiana, Pisum and Oinothera. And two individual chromosomes from different chromosomal pairs to normal diploids are called double trisomy, that means 2n plus 1 plus 1. Tetrasomy, addition of a pair or two individual pair of chromosome to a diploid set, is called tetrasomy, 2n plus 2, and double tetrasomy, 2n plus 2 plus 2. Pentasomy, addition of three individual chromosomes from different chromosome pair to normal diploid sets are called pentasomy, 2n plus 3. Hypoploidy, loss of one or more chromosome from the diploid set in the cell, is called hypoploid. It can be divided into two types, monosomy and nullisomy. And the types of ploidy, there is a flow chart. Ploidy, aneuploidy and euploidy. Up to we have seen aneuploidy. In aneuploidy, it's a hyperploidy and hypoploidy. In hyperploidy, trisomy, double trisomy, tetrasomy, double tetrasomy, pentasomy. Now hypoploidy, monosomy, double monosomy, nullisomy, double nullisomy. And in euploidy, there are three types: haploid, diploidy and polyploidy. And haploidy is a monoploidy, and in polyploidy, autopolyploidy, allopolyploidy. In autopolyploidy, autotriploidy and autotetraploidy. Now hypoploidy, monosomy means loss of a single chromosome from the diploid set are called monosomy, 2n minus 1, and double monosomy, 2n minus 1 minus 1, and triple monosomy, 2n minus 1 minus 1 minus 1. Nullisomy, loss of pair of homologous chromosome, that means two pairs of homologous chromosome from the diploid set are called nullisomy, 2n minus 2, double nullisomy, 2n minus 2 minus 2. Euploidy, euploidy possess one or more basic set of chromosomes: monoploidy, diploidy, polyploidy. Two sets of chromosome are called diploid. Half the number of somatic chromosome is called haploid, and polyploid, it's an addition. it should note that haploidy is different from the monoploidy. For example, the common wheat plant, and is a polyploidy, hexaploidy. 2n equal to 6x, that is equal to 72. It takes place in wheat plant. Monoploidy condition occur when an organism is under polyploidy condition. In a true diploid, both the monoploid and haploid chromosome numbers are same. Thus, a monoploid can be an haploid, but the haploid cannot be a monoploid. Polyploidy, where the conditioned organism possesses more than 2 basic sets of chromosomes. If it is 2 sets of chromosomes, that is called diploidy. Half the number of chromosomes called haploidy. More than 2 sets of basic sets of chromosomes called polyploidy. They are the triploidy, tetraploidy, pentaploidy, and hexaploidy. And polyploidy are 2 types: autoploidy and alloploidy. Autoploidy possess more than 2 haploid set of chromosomes, and there are 2 types: autotriploidy and autotetraploidy. Autotriploidy means 3 sets of the own genome they produce artificially. Triploid sugar beet have higher sugar content than the diploid and are resistant to molds. And seedless watermelon, apple, sugar beet, tomato, banana or man-made autotriploids. Autotetraploids have 4 copies of its own genome. Example: rye, grapes, alfalfa, groundnut, potato and coffee. Alloploidy, 2 or more basic sets of chromosomes derived from 2 different species is called alloploidy. Example: triticale, the successful first man-made cereal. Depending on the ploidy level, triticale be divided into 3 main groups. Tetraploidy, that is crosses between diploid wheat and rye. Hexaploidy, cross between the tetraploid wheat, triticum durum and rye. And octoploidy, crosses between the hexaploid wheat and sativum, triticum sativum. In triticum durum and secale. In this triticum consists 28 chromosomes and secale consists 14 chromosomes. When the gametes are arised, they are in triticum the 14 and secale is 7. And in first F1 hybrid, F1 generation hybrid, 21 chromosomes, it doubled by triploidy, so it's a sterile type, and doubling by with the help of colchicine, it doubled into 42 chromosomes. Then the critical hexaploidy arise. Critical that colchicine is an alkaloid extracted from a root and corm of Colchicum autumnale. When applied in low concentration to the growing tip of the plant, it will induce polyploidy. Significance of ploidy: more vigorous, more adaptable, and many ornamental plants are autotetraploids, have large flower and longer flowering duration. Autoploidies have high increase in fresh weight. Aneuploidies are useful to determine the phenotypic effects of loss or gain of different chromosomes. Many angiosperms are alloploids and they play a role in evolution of plants. Structure change in chromosome: structural chromosomal aberration, addition or deletion of a part of chromosome leading to the rearrangement of gene called structural chromosomal aberration. and this aberration is a change in the number of the gene loci and change in the arrangement of gene loci. Change in the number of the gene loci, deletion or deficiency. Another one is a duplication or repeat. Change in the arrangement of loci is inversion and translocation. Deletion or deficiency, loss of a portion of chromosome is called deletion. Example, Drosophila. There are two types: terminal deletion, intercalary deletion or interstitial deletion. Terminal deletion means single break in any one end of the chromosome. Intercalary, it may break in two breaks and reunion of the terminal part leaving the middle. Both deletion unpaired, it forms the both deletions they are form the unpaired loop formed in the normal chromosome part at the time of chromosomal pairing. Such loops are called deficiency loops. Duplication or repeat. Repeat more than once in the same chromosome is known as duplication. Example, it was reported in Drosophila by Bridge, and another example is maize and pea. There are three types: tandem duplication, reverse tandem duplication, displaced duplication. Tandem duplication means duplication segment is located immediately after the normal segment. Reverse tandem duplication are located immediately after the normal segment, but the gene sequence order will be reversed. Displaced duplication away from the normal segment. That duplication segment is located away from the normal segment. Duplication play a major role in evolution. Inversion, gene in a chromosome reversed by an angle 180 degree. The inversion was first reported in Drosophila. That inversion is paracentric inversion and pericentric inversion. Paracentric, an inversion which takes place apart from the centromere, and pericentric, the inversion include the centromere. Inversions leads to evolution of a new species. Translocation, transfer of a segment of chromosome to a non-homologous chromosome is called translocation, and which is differ from crossing over. In crossing over, exchange of genetic material between homologous chromosome, but in translocation between non-homologous chromosome. There are three types: simple translocation, shift translocation, reciprocal translocation. Simple translocation, single break is made in only in one chromosome and get attached to the one end of the non-homologous chromosome. Shift translocation, broken segment of one chromosome get inserted intricately in a non-homologous chromosome. Reciprocal translocation, exchange of chromosomal segment between two non-homologous chromosomes. It is also called illegitimate crossing over. There are two types: homozygous translocation, heterozygous translocation. Homozygous, the chromosome of two pairs are involved in translocation. Heterozygous translocation means only one of the chromosome from each pair of two homologous are involved in translocation.